Hi, this is Dr. G. Bailey, and this is topic 1.6. What I'm doing here in this video is going through long and short answers related to 1.6, which is all about mitosis and cell division, cell cycle. So let's go ahead. So one of the first questions that is most commonly shown, and by the way, these questions are coming from past IB questions papers. So one of this is talking about cell cycle. So here, cells go through a repeating cycle of events in the growth regions, such as the plant root tips and animal embryos. So sometimes IB will come up with a whole sentence. You're reading this and saying, what's the point of all that? And then eventually it gets to the real deal, which is the right question, which is outline the cell cycle. So you might think, well, what do I do about the first sentence? Nothing. It was just an introduction. So here it says outline the cell cycle. Now, I'm going to do quickly on what the cell cycle is as a refresher to your mind. Cell cycle is how the cell is living his life. So usually the cell cycle lives in G1 when he's growing, developing his life, and then he goes through S. Now, he only will go towards the S if it's time for him to divide. He realized that the cell gets too big, right? The cell got too big, so I think it's time to divide. So what he does is at S, he goes to replication and makes a copy of himself. After he finished copy, he moves to G2. When G2, the cell continues to grow, and at the same time, you're making extra copies of the organelles. After they're ready, right about this point, when they're ready to divide because they have copies of everything, they enter mitosis. Now, in mitosis, we know that there's 4 stages, right? And you have to be able to explain those 4 stages, or at least name them. So the first one is prophase, metaphase, anaphase, and telophase. So these are the 4 stages of mitosis. Once mitosis is over, then comes the next stage, which is the cytokinesis. Now, kinesis, just for you to know, kinesis, it's completely different between the animals and the plants. In animals, it's cleavage furrow, while in plants, it's cell plate. And once you finally get 2 cells, these 2 cells go back to living on G1 for a long time, grow, develop, and that's the cell cycle. Out of the cell cycle, there is the interphase stage, which is just G1, S, and G2. These 3, they make up the interphase, which interphase is just an area for most of the growth and development and living. So now that we got a very quick review on this, if you need to know more details, go back to the lecture. Now let's go to the mark scheme. Look in the mark scheme. The first thing that they ask is describe what mitosis is. Mitosis is the division of nucleus to produce 2 genetically identical daughter nuclei. So if you just say mitosis is a cell division to get 2 daughter cells that are exactly the same as the parent cell, you're good. Next, what does he have? 4 stages: prophase, metaphase, anaphase. Next, then after mitosis, cytokinesis occur. What comes after this? Then you enter the interphase, which is basically the metabolic after phase. You can also say the enters interphase, which we have the stages of G1, G2, which is here. Enter the phase of S phase G1, G2. Now, in this mark scheme, it's not in order, but it's G1, S, G2. But if you're just saying that interphase has these stages, you're still fine. Also, what happens next? It actually goes to DNA replication. And then what you got to talk about is that there was a cell growth or preparation of mitosis or duplication of organelles in G1, G2. So we're talking about G1 and G2, which is all about growing, and G2 is all about preparing the organelles. So this is what the answers will be, because what is this describing? It's describing the cell cycle, right? And it's about outline. So when it says outline, you don't have to describe prophase, metaphase, anaphase, and telophase in details. You don't. It's an outline. So you're just outlining what he has. And outlining means mention interphase with the 3 stages. Talk briefly what each stage does, what S does, what G1 does, what G2 does. Mention mitosis, what is it about? What are the 4 stages? And then cytokinesis, what is it about? You probably will hit this outlined cell cycle, usually would be something like 4 or 5 points. And you can see that there is more points in here. So this is a very common. If you'll be able to describe these stages, then you're good to go. Another type is about cell cycle is explain how the cell cycle is controlled. Now, Cell cycle controlled is not the same saying what is the cell cycle. The cell cycle is the stages we're talking, but the question is control. Control means when do we go from G1 to S, and when do we decide to leave S and go towards G2, and when do we leave G2 to go towards mitosis, and when do we finish mitosis and go back to G1. Now, to do this, it has to be regulated by proteins. In this case, we have a protein named cyclin. And when it comes to cell cycle, there is 4 of them that you have to know, which I usually say is DEB. So cyclin D, cyclin E, cyclin A, cyclin B, these 4 proteins regulate it. What do? Usually a cyclin, like this little circle here, will activate an enzyme. And the name of this enzyme is known as CDK. So every time the cyclin touches it, it activates it. And when it activates, it moves to the next stage. But you have to have a lot of it. So how does it actually work? So when you're going from, for example, from G1 to S, you need to have a lot of E, a lot of E. When you have a lot of E, eventually you will activate CDK, and now it's time to move to S. Now you are on an S. When it's an S, you have to produce a lot of cyclin A. When you start producing a lot of cyclin A, you reach a point you activate the enzyme CDK, and now you move to G2. Once you get to G2, now you're producing a lot of cyclin B, and you're producing a lot of cyclin B, and it's just building and building and building to a point it is too much that it activates the enzyme CDK, and now we move to mitosis. Now, when you want to get mitosis back to G1, that's when you produce the cyclin D, and then it activates the CDK to go back to G. So it usually is known as a DAB, and you can start from mitosis to G1 as you want, but this is the pathway to memorize DAB, okay? So basically is what it's saying. You need this protein to be in a large amount to move to the next stage. Otherwise, it won't work. So if you have just a little bit of E, you will never go to S. If you have just a little bit of A, you will never go to G2. If you just a little bit of B, you're not going to go to M. If you have just a little bit of D, you're not going to go back to G1. So the protein cycling is very important in the shifting of one stage to the next. So this was a quick review how it works. Now, of course, there's more information to this. You can always go back to the lecture, which we talk about the CDK and how the CDK can actually cause phosphorylation, which means the adding of the phosphate to activates other stuff. But let's get to the mark scheme. So one of the first things on the mark scheme is cell cycle is a sequence of stage. So what is it doing here? First, always introduce what the hell is cell cycle. I always tell students the first answer that you have for a question is define what it is. If it's a cell cycle, define what cell cycle. If it's mitosis, define what's mitosis. If it's a hormone, define what a hormone is. Next, what is the cell controlled by? Cyclins, right? We have to mention it. Next, levels of cyclin. Remember, it does fluctuate during cell cycle. What does that mean? When you have a large amount, then you move on to the next cycle. So talk about that. Then condition inside as well as the outside affect the regulation. Now, this is from the outside. Like if the environment is saying, hey, we need to repair cells, so I need to do more cells. Or let's say the cell is getting too big. If the cell is getting too big, then you have to divide. So here's just saying, when would you divide? It depends on the conditions of the cell or over the surroundings of the cell. Next, we talk about the 4 cyclins. So you can talk about the DAB that I just said and how it regulates. You can talk about their sequence, the timing, right? How the cyclin triggers the next stage. So talk about the DAB. And then here, the idea of different cyclins acting in different phases must be clear. So what does that say? You have to remember, mitosis to go to G1, you need D. G1 to go to S, you need E. And then S to go to G2, you need A. And G2 to go to M, you need B. And then you just go back again up here. So you have to be clear and mention these stuff because you can get points for showing this. Now, the upper part here, the next part where it says here, let's put it in green, cyclin-dependent kinase, which is CDK, binds to it. So here, You don't have to know how to write cyclin-dependent kinase. If you write CDK, IB will still be okay for the SL students, but if you are HL, you should be able to write cyclin-dependent kinase, okay? That's the enzyme that the cyclin will bind to it and move to the next stage. Now, what does this enzyme do? It's a kinase, which that means it phosphorylates. That means it adds a phosphate. Oops, let me put it here. It adds a phosphate to another molecule or to a protein. So that's what kinases do. They add phosphates to something else. And what it does, it makes the proteins being phosphorylated, and that means they do a certain function in the cell cycle. But you don't need to go that detailed. This is more like an HL situation, but it's barely seen in the mark schemes, but it's seen in this one, but I just added for the sake of it. But if you have from this point up to here explaining the four cyclins and their levels, then you probably got the full points. Okay. Oh, forgot the F. Also, you have to mention the CDK. You will get 1, 2, 3, 4, 5, 6, 7. It's exactly 7 points. Not this one, but you can talk about the DEB. If you put the DEB, usually in the mark scheme, they will give you those answers. So once again, explain what cell cycle is. Talk about cyclin being the one that regulates. Talk about how it touches the CDK, which phosphorates other proteins. Talk about the levels of these cyclins on how you go from one stage to the next, and how the levels of these cyclins influence the whole thing. So these are how you're going to explain the entire cell cycle. But I've seen many questions like this one, but usually they're not more than 4 points. This one, for example, it's 7 points, so I put the maximum you can get in there. But usually I see more as a 4 or 5 points, so you can easily nail it on this question. Now, the next question is distinguish between the outcomes of cell dividing either by mitosis or meiosis. Distinguish, usually it's up to 2 to 3 points. It's like, go brief on it. And the word distinguish here, right, this means what is the difference? What are the difference between mitosis and meiosis? So the difference between these guys is that this one, you want to produce sperms and eggs. And since you want to produce, this is basically, this is actually sexual reproduction. This one, you want to produce identical cells, right? So this is more asexual. And then in mitosis, you have one cell that will become two, and these guys will be identical as the parents. While in meiosis, you start with one, you go through a first division, and then you go through a second division, so you end up with 4 cells at the end. And that's the differences between the two. Now, these 4 cells that I'm talking about here, it can be 4 eggs or 4 sperms. And these guys are just replacements. Let's say you want to make a new bone cell, you want to make a heart cell. It's just replacing ideas. So now that I can give you a little bit of a scoop, let's go to the mark scheme. The first thing it says is mitosis produces 2 daughter cells while meiosis produces 4. Just like in the pictures right here, as you can see, this produces 2 and this produces 4. Next, mitosis produces cells with the same number of chromosomes while meiosis produces with half. Now, this is basically saying that mitosis, right, with the 46, the final cells will come out to 46, which is just like the parents, right? While in meiosis, if you have a 46, the whole process of going through 2 cells and dividing again is that in the end, all those 4, they will have half of it, 23, 23, and 23. So although this meiosis is topic 3.3, you will see them mixed. But if you want to learn more about meiosis, you can go to topic 3 to get a little bit more on the lecture on this. Next. Then we have here, mitosis produces body cells, while meiosis produces gametes. And the word gamete here is talking about sperms and egg. Just a little bit of a vocab, if you say the word somatic cell, this basically means body cell. And if I say gametes, gametes means sperms and eggs. So somatic cells is like your heart cell, your lung cell, while gametes is basically sperms and eggs. So you basically saying mitosis produces these guys while meiosis produces these guys. Okay, now allow answers to be in table, reference both that has to be present in the mark. So basically you can do as a table. One side mitosis, meiosis. So it's a table like this, and you have mitosis here, and you have meiosis, and you start mentioning them. So this is the question for this. Is very brief. Now, this next question is talking about how to outline the process that occur in cell during interphase, including those needed to prepare for mitosis. So it's the same thing talking about the cell cycle, and I'm not going to go through the story because in the beginning of this video I explained it. So let's go straight to the answer mark scheme. But before we get there, let's clear again. It says outline, right? What it wants, the process that occur where? In the cell. And it says just during interphase. This is where students get it wrong. It's just during interphase. And what is it that they need to prepare for mitosis? So it's just talking about G1, S, and G2. It is not talking about mitosis process. It's not talking about cytokinesis. It's just about these three. So talk about what happens in each of these three. Obviously, you can have S, which is DNA replication that is happening. It also has DNA transcription because it's true. During G1, you can have transcription and translation happening, which that means you can do protein synthesis, biochemical reactions. That's what happens in G1. Cell respiration happens during G1. You have growth, of course, G1, G2, the cell continues to grow in size. And in G2, you have organelles replicating. So just a real scoop. Remember that G1, these cells are living. And when the cells are living, they're living every day. They usually are doing these protein synthesis. They are doing, for example, a cell in your stomach is going through cell respiration in the lungs. It's working, whatever it is. So all of these guys is G1, G1, G1. This is G1 and G2, and this is G2, and this is S. So this is describing the things that is happening to the cell as it goes through interphase. A lot of students think that G1 just means growth and development. Well, what does that word develop means? It means transcription translation. It means chemical reactions. It means cell respiration. Growth. The cell will continue to grow as you exercise. So it's totally fine that you can mention G1 or G2. But the place where organelles are actually duplicated, that's only at G2. And where the DNA is replicated, it's only at S. Make sure you know that. So for the next question, explain the reasons for cell division in living organisms. Now, here you got to be very careful. It explains the reason. So why? Why do we need to have cell divisions in living organisms? It's not asking you to describe mitosis. It's asking, why do we have mitosis? What's the purpose in having mitosis? So with that in mind, so for example here, explain the reasons for it. The first thing you would say is to increase the number of cells in an organism. That is true. Use the logic. If you start as an egg and a sperm, right? And they come together, boom, you have a zygote, right? Or a fertilized egg. Now what happens? This is you. Smiley little face. Woohoo. You're going to stay as a cell? No, of course not. What are you going to go through? You're going to go through mitosis. Why? Because you're going to become a bunch of millions of cells, and then eventually going to become this huge person here with trillions of cells. So of course, you have to increase the number of cells of an organism. It allows differentiation and cell specialization. It goes back to the idea that if you have here as a one cell, how do you want that cell to become a heart cell, a lung cell, intestine cells? No, you can't just have this one guy being all of this. So if you make more cells, you can dedicate these guys to be a heart, and if you produce more cells, you can dedicate them to be a lung. So it allows you to do differentiation, to give like cells, each one their own function. In this case, we use the word specialization. For greater efficiency, yes, the more cells you have, the more they can work together, right? That's how it is. To replace damaged cells. So let's say if you burn a little bit of your skin, you always can replace that skin. But how are you going to replace? Obviously by making more cells. If you want to make more cells, you have to go through cell division. Binary fission. Now, interesting that they just have the word binary fission here. which people can say, how is this a reason? Well, again, IB sometimes is just throw words. You can say binary fission is for bacteria, while you might have mitosis for, animals and plants. And they were like, yeah, get your point. Weird? Yes. It's IB. Now you gotta say asexual production for unicellular organisms, which that means cell division when it comes to cells that are just made of one cell, right? Like Paramecium, it's just one cell, it just goes through one division. Continue. Gametes, spore formation. Even if you want to go from one egg, one cell to make sperms and eggs like this one, what happened? They had to go through cell division to reach to the point where you're producing those sperms and those eggs. You have to go through it, right? You cannot just go from one cell to make it become a sperm like that. It had to go through cell division. That's why you need it. Also, cells only rise from pre-existing cells. This is the most obvious that people don't say, oh my God, seriously, I have to mention that? Yeah, like how the hell did the cell come from? Obviously from a previous cell that went through division. Otherwise, you don't get that cell. What are the reasons for cell division? Cells cannot grow beyond a certain size. If you have learned about surface area to volume ratio, you know that the cell reaches a point where it cannot function, so it has to divide into two. So this is again, the surface area ratio becomes too small and we need to move on. Also, we have transport across the membrane is too slow. That's what I was talking about. If the cell gets too big for this guy to leave, it's going to take longer. If he was this tiny, it goes quickly out. So again, if the cell gets too big, it has to divide. Of course, the nucleus cannot control the cell if it's too big. That's why you have to divide. And control the cell division sometimes is lost. Now, if that happens, look how this is crazy. It says here, explain the reason for cell division. Now, cell division is not saying always the healthy way. You can say sometimes there is cell division, and why? Sometimes the cell division can go like so quickly and uncontrollably, like it's just dividing so fast. And when this happens, usually it has to do because you lost the control of cell division, and that led to what we call tumor formation, which can lead to cancer. So these are all reasons why cell division happens. And remember, it's not always a good thing, like cancer, tumor. A lot of cell division, it can be bad, but that's what happens. It just means that the nucleus lost control of the cell division. Now, refer to Virchow, you don't have to know about this one, but this is it. So this is the things that you need to explain about cell divisions, the reasons, okay? They're growing, they have to divide, or you want to make more of a certain type of tissue, or you want to replace a tissue, or you want to specialize more cells into a certain thing, so these are all types of reasons why you want to go through cell division. Next question, it says here, outline what is indicated by the mitotic index of tissue taken from a tumor. This is outlined, right? So it's basically in descriptions. Basically, they're saying, what does the mitotic index tells it? So here is just asking you to say, what's the purpose of mitotic index? And if you remember, mitotic index is to check if the person has cancer. And usually what they do is they take a tissue, like you can see here, they take a tissue and they look in the microscope. They count how many cells there is. Let's say there's 100, and then they count how many cells here are going through mitosis. Let's say there's 50. That's 0.5. So that means this person has cancer. If the person had 40, then it would be 0.4. That person doesn't have cancer. If that person has 60, which is above 50%, 0.6, that means the person has cancer. That's the point of mitotic index. So if I had to look at the mark scheme, it says mitotic index is an indication of a ratio, which is true. We saw the ratio of the amount of cells going through mitosis over the total. So, or just says the percentage of cells undergoing mitosis. Also, cancer cells divide much more than the normal ones. So we can talk about cancer, how cancers means that when the cells are dividing uncontrollably, so you have a lot of cell division happening. Another one, a high elevated mitotic index in tumors. That's why I told you here from the beginning that if you have a big number, anything that is above 0.5, above that, it means that you have cancer because it's more than 50%, right? It has to be above more than the half. So 50% of 100, that means that is more than the half. Most likely they have cancer. So that's what this thing was saying. You have to measure how aggressive it is by seeing the elevated mitotic index. So, I mean, the bigger the number of mitotic index, more likely you have a tumor, more likely you have cancer. So that's how you would outline. You just describe the equation of mitotic index and talk about cancer. What is cancer? It's just a bunch of cells dividing uncontrollably. And that's pretty much for the topic 1.6. But here are some extra questions that I barely seen in the IB past papers, but it could be coming. Like the first one is the most interesting one, is describe mitosis stages. I haven't seen them put this on paper too, but I have seen a lot of multiple choice questions, like identifying basically the micrograph, but describing every single stage, very rare to see, but it could happen. Also, talking about what is oncogenes, which is cancer genes, metastasis, malignant tumors. These are all in my lectures for 1.6. Distinguish between what is chromatin and chromatid, or what is chromatid and chromosome. Explain how CDK enzyme is important in cell cycle. Describe cytokinesis in animal plants. You have to know what's the difference between the two, and I haven't seen a question on IB past papers asking about it yet. Which stage in cell cycle is the longest, which is G1. If you could add meiosis stage into the cycle, where would you locate it? And it's usually the same place as the mitosis. You just have to annotate it. And which stage of cell cycle does transcription translation happens, which is G1. So all of these questions are possible future questions that might file an IB. And these are the ideas that came into my head. But you can find these answers easily on the lecture of 1.6. And that's it for this topic. Thank you.